Go for it.
Welcome everyone to a Systems Neuroscience and Complexity seminar. Today, I have the absolute great pleasure and honor to invite Eli Muller, who's a really talented postdoctoral scientist that I've been working with and I really feel collaborating with for the last few years. Eli, I was actually introduced to him by Ben a number of years ago when I was starting out my lab, and I knew at the time that I really wanted to embrace the kind of power and benefits of computational modeling and a kind of deep understanding of the mathematics of dynamical systems, complex systems. But I also knew from my short interaction with that field just how difficult that world is, how many pitfalls there are, how many ways there are to do modeling in ways that they're almost a little bit like fool's gold, where you feel like you've got an answer, but you've got an answer for a reason you didn't anticipate. And the results are actually then, they appear as though they're meaningful, but then they actually fall out from underneath you as soon as you change some parameter or as soon as you actually check your assumption. And so I knew very early on that if I was going to do that kind of work, I needed to find people that I could work with that could do that work at a high level. And also really listen to the kinds of things that I would whinge and whine about, the kinds of neuroimaging things that were important to me and the kinds of neurobiology. And I thought, at the outset, okay, I'll find someone who's got a background in this stuff and this would be great and they can do some work. What I didn't anticipate, and the part that's just been absolutely one of the best things that's happened to my career, is finding someone that listens carefully to what I care about, but then advances it and pushes back against the things that I wouldn't want to work on and makes me ask my questions better, makes me think more deeply about what I'm trying to achieve and why I'm trying to achieve it, what kind of answers I'm looking for. And so Eli's been a huge, huge push in that regard. Working with Brandon as well has just even made that more satisfying and enjoyable. And I can't wait to see what we come up with next. And I'm really excited for you to all see what Eli's been working on, what he's thinking about, and to listen very carefully to where he can see from his amazing vantage point that he's been working so hard to create. So excited to see you talk today, Eli.
Goodness gracious. Thank you, Mac. As you can tell, Mac and I have a serious bromance problem. Everything you said is very much mutual, and it's been one of the great friendships and working relationships of my life. So I very much enjoy working with Mac, and like he says, working together to shape each other's thinking and embracing differences in perspective and really trying to resolve incongruencies when and where we find them. It's been super satisfying for me as a scientist. All right, so I guess I will start my talk. Let's hide everyone. It's the worst bit about doing Zoom talks. You can't have anyone's faces on this. I'm talking to my slides. It's horrible. Anyway. All right, well, thank you everyone for tuning in. It's going to be a little bit of I'm not going to say tour de force. It's not a tour de force. We're going to go on a little bit of a tour that follows along with some discussions Mac and I have had over the years, and sort of as we start to expand our thinking on the various projects that filled in, as we started to develop an understanding of what we were really going after. And it all sort of spawned from an early discussion I'd had with Mac. He was teaching me a lot about neuroanatomy, of which I proclaim to have very moderate understanding of. I come from a physics and mathematics background, but very interested in biology, and Mac was really educating me on different circuits and connectivity profiles and what have you. And we had this wonderful discussion about diffuse connections in the brain and what their role might be in the broader organization of neural dynamics. So that's what we're going to be talking through today. Let's just get this one out of the way first too. I'm currently at Sydney University, although I am from New Zealand. If you can't tell by the accent, it looks something like this. I actually live about 45 minutes drive. Well, I grew up 45 minutes drive from Hobbiton. So in a very real sense, I have, just like Fredo and Sam, departed the Shire and wandered out on to the broader Middle Earth. Hopefully it doesn't terminate at Mount Doom, but in this analogy, Mac has already painted himself as Aragorn, son of Arathorn, but we won't let him have that one. So as I said, today I'm really interested in how the brain organizes across scales, and indeed our lab more generally is. And the interesting questions for me are, we've got these electrically active cells in the brain. They fire based upon the inputs that come in, but somehow together they are able to perform complex cognitive tasks, behavior, and indeed they organize in a certain way to achieve this. and really tapping into what that organization is and what are the principles that govern that organization is very much an interest of mine. I'll just quickly whiz over this, but one of the issues with like resolving this mesoscopic scale is we have these observation keyholes into what's actually going on in the brain. Now, these can be spatially or temporally constrained, like you might only see a part of the system, or maybe you see the whole system, but really blurrily. And indeed, there's extra biophysics and biology that sit on top of these. So these modalities that are tapping into the dynamics of the brain, neural activity, are actually tapping into an aspect of the physics or the neurobiology. So they need not, just because they overlap in this spatiotemporal diagram here, it doesn't necessarily mean they're tapping into the same type of quote unquote activity. They're measuring something, and then you need some type of observational model that can kind of relate them all. There's some fMRI and human and macaque. And we also have, which is measuring blood oxygen level dependent signals. So it's sort of trying to capture this idea that neurons, when they're electrically active, are consuming oxygen. And then you can tap into that oxygen consumption as a signal and infer neural activity. In parallel, there's the electrical measurements that we make. You can either have it with depth electrodes, measuring things like local field potential, so the changes in electrical activity, some sort of localized region of the brain, as well as magnetic signals that we can measure with MEG. And then on top of that, we have an analysis keyhole into dynamics. Just sort of we're at this point, we have to choose the way we analyze our data, and we can do that by making assumptions about the brain being static and looking at things like correlation, or maybe we just measure white matter fiber densities between brain areas. Maybe we're more focused on statistical measures as opposed to generative measures. So starting with like, this is the biophysics. What would that look like as a statistic if we ran some model of that biophysics? And equally, we can address local or global scales. So somewhere in this diagram, we wander around as we're doing our particular analyses in our particular data set. But they each have their own bias. They provide a keyhole into what's actually going on, the complete dynamics. And then within that space of uncertainty, I'm very much interested in applying dynamical systems theory, in large part because I come from a physics background, using things like attractors and bifurcations to provide intuition about how this really complicated system is organized. Like these provide a low dimensional description or understanding of how some particular dynamical system operates more generally, but can we apply them to the brain in meaningful ways? I mean, the truth is they work just like some of the techniques we've seen in this diagram, where you can apply them in many different ways, right? Like it's true that a neuron has an attractor. It's true that neural systems have attractors. So depending on how you apply this, you'll get different answers. So yeah, I think a lot about meaningful ways to apply this type of perspective. All right, for the precursor stuff out of the way, let's dive into it. So diffuse brain systems from theory to observation and back again. That's the last Lord of the Rings joke I'll make. So it turns out that there are lots of diffuse systems in the brain. The ascending arousal system contains a few of them. One of the key ones is the locus coeruleus here. I don't know if you can see my cursor, but it sits in the pons, projects noradrenaline really broadly throughout the cerebral cortex. So a particular cell within the locus coeruleus may, and it need not be synaptically, it may be extrasynaptically, and indeed it likely is in a lot of cases. It's depositing noradrenaline throughout the cerebral cortex and changing the neural dynamics there. But like, what is that doing? And like it goes beyond this traditional story of neurons sending messages between each other, right? This neuron fires. There's some downstream neuron here that waits, and if it hears the little ring from the neuron upstream, it then activates itself. This sort of control perspective perhaps might be more appropriate. It's just another diagram here. There's also other systems that aren't in the brainstem, like the thalamus, that have diffusely projecting profiles to them. Here's an awesome diagram and a wonderful paper from a wonderful paper by macaque and others, showing that even within the thalamus, there's this rich neural diversity where some neurons project targetedly to the cortex. So they, a particular neuron in the thalamus projects up to the cerebral cortex and just in a really localized part of it. And the traditional story of the thalamocortical interaction is that topographically maps back to that same area of the thalamus. So you get these sort of closed thalamocortical loops that are formed. It turns out that that's only true for some of the cell types, and it's actually a much blurrier picture. But there is a neuron, there are nuclei that sit at the other end of the spectrum. So rather than going to some targeted part of the cortex and mapping straight back down to it sends out projections to really broad parts of the cerebral cortex, much like the locus coeruleus projections. Although here, again, it's a bit of a murky story, but it's true. It's synaptic as well as extrasynaptic. And indeed, these particular thalamic nuclei are targeted from different subcortical structures like the cerebellum and the basal ganglia. That's a long story for another day. So really, I just want to start from something really simple. What could diffuse connectivity buy you? Like, why would the brain have it in the first place? Yeah, you can do all sorts of complicated things later on once you accept the fact that it has it, but to first order, what does it buy in the system? And how does that sort of benefit fit into the sort of organizational principles that the brain operates around? So the first thing that Mac and I decided to really tackle was, in the simplest sense, what is diffuse connectivity doing at the large scale? And to do this, we're going to leverage some biophysical modeling, and in particular neural field theory. I won't dwell too much on the math of it all, but the long and the short of it is, the idea comes from statistical physics, where we have a really high dimensional system, like a gas, for example. In a room, there's molecules whizzing around, there's trillions and trillions of molecules whizzing around. And you want to be able to say something about how that gas will operate in certain environments, like when pressure changes or temperature. And that low dimensional description, like picking a number, the average temperature in the room went up, is related to field theories in general. So neural field theory is just taking that type of dimensionality reduction technique and applying it to the brain. And the long and the short of it is, this is something we should all be familiar with. It's just an activation function for a neuron. So as we increase current into the dendritic tree of the neuron, at some threshold, the neuron begins to spike, and then there's a monotonic increase in spike rate as a function of further increases in current. And then we make the assumption that you have some probability distribution of, let's say, spike thresholds. And I say let's say because it need not be spike thresholds. There's some distribution of parameters, and it's Gaussian. You can equally get to this probability distribution by making an assumption about independentness. So if you look at spikes of neurons within some part of the brain, they turn out to be very uncorrelated. And if you have a system of uncorrelated random processes, they converge to a Gaussian distribution by the central limit theorem. They're uniform processes. Some of them do anyway. And, okay, that out of the way, basically you just do a convolution of this activation function with a probability distribution, and you get out this population activation function. So now, rather than focusing on what every neuron was doing, we make an assumption about the distribution of spike rates or something like that, and we get to this mapping function, which means we only have to track the average firing rate of the population and the average membrane potential, and that's sufficient. And everybody else will kind of follow in step, minus some sort of statistical fluctuations around that mean. The mean is sufficient to track a large proportion of the dynamics. I won't walk through the math too much, but there's quite a bit of it. And these are just general functions you see within neural field theory. So like the sigmoid here is defined in the first line. It maps average firing rate to average voltage within a population. Now that can be done in space and at a particular point in time. And then that average firing rate generates a field of activity that propagates, very much like water waves. So here we just you're adding extra activity at a location, like dropping a pebble in a pond, and that activity propagates outwards. You can define that propagation as a wave equation if you so choose. If anyone's read a Nature paper, you'll see that turns out to be a useful thing to do. Synaptodendritic dynamics in neural field theory are pretty straightforward. You just have postsynaptic potentials, these VABs, and they get integrated within the dendrite to cause a particular population's average voltage to change, and that average voltage then induces an average firing rate change, and the whole thing is this closed sort of loop that propagates the activity. And then those postsynaptic potentials also get filtered. There's a temporal filtering effect that goes on as spike volleys arrive into the dendritic tree. They get temporally smooth. And this is true at the population level as well as the single neuron level. And then there's a diagrammatic explanation there showing if you change the timescales of this filtering, you can make the population more or less sensitive to the temporal coincidence of spikes. Okay, so that's all the math and general field theory stuff out of the way. Back to the original question, which was, what's to the first order, what's the main effect of diffuse connectivity? To do this, we leveraged an existing biophysical model. So this is a corticothalamic model that made the that has that really tight topographic mapping of the corticothalamic loop. And now we're going to add in this extra rule of organization, this diffuse connectivity. It's a spatial model, so I had to add in some type of local connectivity. To do that, I just did nearest neighbor in a way that orients the model on a torus. So basically, the brain has these periodic boundary conditions to it, so there's no border of this model. And then the only parameter I change in this work is the diffuse connectivity, the idea that a particular node can send activity everywhere in equal proportion. And that's the dial that I'm going to sweep in my model simulations. So what does the diffuse coupling do? Well, the hypothesis here was something similar to turning up average temperature in a thermodynamic system. So, for example, a glass of water sitting at room temperature on your desk experiences evaporation. The reason this is because there's little statistical thermal fluctuations of the molecules. So their velocities are changing in time as they bounce into each other. Maybe they get collided from all sides and they gain a huge amount of energy. So to a subset of the glass of water, there's a bunch of molecules that might see the average temperature of the water being much hotter than in fact it is if you took into account the complete system. And that means that they're near this phase transition, it's called in physics, where in this case, it's a point with which there's too much energy in the system for the molecules to exist in their liquid phase, and they'll transition into a vapor phase. And that bifurcation, as it's called, can occur for a small subset of the system, while the whole system itself is in fact below boiling point. So much below 100 degrees Celsius, but there's in some sub part of the system, it's sufficient for evaporation to occur. So the intuition here is that the brain might leverage this, like a feature like this, where it can push parts of itself towards this critical boundary, gain these extra features, which, in the case of the thermal system, isn't anything particularly interesting, but in the case of the brain, I'll show you, are actually quite rich. Here's me double clicking on the thermal system, where basically these curves show the spread of velocities for the molecules within some, I think this might be water. I can't remember exactly what it is, but the A parameter here is the average temperature of the system. So you can see here the velocities, there's no one below zero, and everyone has a velocity that's above zero, and it has some Gaussian-like distribution. And as we turn up the temperature, you can see not only does the average velocity of the particles increase, but so too does their spread. So there are some particles that are going really fast and some going really slow. That's important because you're promoting heterogeneity in the system just by turning up an average temperature dial. So the intuition here is, this is a normal bifurcation diagram you might see with multi-stability. The brain and all of its nodes are these little white circles here. And in the subcritical regime, they're all really homogeneous. They're sitting around some sort of local attractor. And as we're turning up this diffuse connectivity dial, we push the system collectively towards a bifurcation. But some of the nodes can get closer than others, because as we're turning up the dial, like I said, you get much more of these extreme conditions in the system. And indeed, some nodes are still really subcritical and some are much closer to the critical boundary. So let's get into the brass tacks of it. We can actually go through and calculate this explicitly in the model. You can go through and work out what the attractors are. So you solve for the steady states, and you can sweep the input of each node in the system. So you just make a first order assumption. You say, you know what, the network's doing a bunch of interesting stuff, but I don't really care about what the dynamics of it are. I just care that it's giving me an extra bit of current. And we sweep that current parameter before running any simulations, just to say how much current would each node need to get across its bifurcation. Then when we go and run the full simulation, we can say at this point in time, this node was this close to its bifurcation, this node was this close, and so on and so forth. So we get a real-time measure of how critical each node is. And you can see here as a function of diffuse coupling, as we start to turn it up, the nodes, more and more of the nodes start to cross that bifurcation. and eventually, in the extreme case, all of them cross, because the scaling of connectivity in the system is too large that no node can sit below this critical boundary. So that regime here is called the quasi-critical regime. The idea being that you don't actually want to sit in this upper bound where most of the nodes have crossed. You want to sit somewhere down the bottom where only a few of them are getting across, and they're forming these like quasi-critical assemblies. Another way of viewing it, this is functional connectivity along the top here, so correlation structure, and you've basically turned the correlation structure into what's called a like a force-directed plot. Essentially, each correlation is worked out as effectively like a spring. So each node has a spring to every other node. You do a bit of thresholding here, but the long and the short of it is that the nodes that are really strongly correlated have a strong spring, and you just do this little graphic here that shows the system on the left here, when the diffuse coupling parameter is low, has organized to the toroidal structure, the structural connectivity that are baked into it. So that nearest neighbor connectivity is dominated. And as we turn up diffuse connectivity and we move into the quasi-critical regime, you can see we get this really interesting heterogeneous organization. Some nodes are really low dimensional in their interaction with the network, and others much more high dimensional. And then in the extreme case, it's just a spider's web mess of everyone's kind of not listening to anyone. You can go through and ping the system to observe how it responds to stimulation. And you can see on the left in the subcritical regime, it's really homogeneous. Everyone has the same sort of time scale of response to stimuli. And as we move into the quasi-critical regime, like I said, we get this heterogeneous response. So now there's a heterogeneity of timescales, so a gradient of timescales. And that's because what I'm showing on the bottom here, you have an attractor, and as we turn up the diffuse connectivity, we're flattening that attractor landscape, and the system can now have long excursions away from this little minimum in the energy landscape. And in the extreme case, that attractor is completely lost and it rolls away to this upper attractor, which in this model is something like a seizure. So do we see evidence of this type of attractor landscape flattening in neuroimaging data? This is some awesome work led by Brandon Munn, that basically we took fMRI data. We were able to isolate voxels in the locus coeruleus, so that brainstem structure that I mentioned at the start that projects noradrenaline really broadly throughout the cerebral cortex, as well as a competing cholinergic structure called the basal nucleus of minute, which does go to lots of different cortical areas, but in a really targeted fashion. And in fact, acetylcholine does something sort of opposite to what the locus coeruleus is doing, where the locus coeruleus is looking to co-activate multiple areas and bind them together. The basal nucleus of minute is really constraining and stabilizing local dynamics by trading off the network terms. So taking fMRI data, we're able to go through and calculate an energy landscape. The math of this is not really too important here. The intuition really is the idea that at a particular point in time in your time series, you can say, I'm here in my state space. And then in the next few time points, how quickly do I move away from where I am right now? And you can do that for every time point in your time series. And you can generate a probability distribution to say, how likely was the change that I saw? Because maybe some points in time you run away really quickly from where you were, and other times you hang around there for ages. And we want to quantify that in terms of a probability. Once you have a probability, you can turn it into an energy. And that's what you're seeing here. So mean squared displacement's a measure of how quickly you ran away. Energy is obviously to do with the probability, and then time is in the next few points in time. So there's some more intuitive labels that are coming up here. So likely, unlikely, soon and later, and near or far. That's a Celine Dion joke if you want to say one too. But yeah, so essentially when this is really flat for large displacement and for a long time after a particular time point, that in this case, that's quite likely for this for these data. And if you compare it to periods where the locus coeruleus activity was high relative to the basal nucleus of minute and the inverse as well, you find that when the locus coeruleus was activated, this diffuse structure, it's much more likely to find large changes in your time series for at all sort of like for early and for late time periods. It's because the energy has dropped in this energy landscape here. So this flatter area here is basically saying it's much more likely to find large changes in your system. And conversely, when the basal nucleus of minute is activated, it's much less likely relative to baseline. So that's good. that's making intuitive sense, and it overlays with what we were finding in that really vanilla version of diffuse connectivity we were showing in the model previous. What about causal evidence? So there's some awesome work from Valerio Zerbi's group with mice where they were doing optostimulation of the locus coeruleus. So really getting, they genetically modify the cells within the locus coeruleus to respond to a particular frequency of light. They put this optode in there and they fire in that color of light and they activate those cells. And what they find is when they activate the locus coeruleus, the cortical dynamics flatten. Doing that exact same calculation I showed you in human fMRI, we do it on the mice, and we find that the energy landscape is indeed flattening when we stimulate the locus coeruleus. So that's cool because one wasn't causally influenced, it was just inferred based upon its current dynamics. And then this follow-up work says when we actually causally manipulate it, we do indeed see the flattening. But what about the influence of these diffuse connections on stimulus response? So for that, we're going to jump to another species, which is the larval zebrafish. For those who don't know, there's some awesome calcium imaging work that goes on in this organism. They're able to capture upwards of 40,000 cells, the calcium signals from 40,000 cells, just getting up, greater than 60%, in some cases, much more coverage of the zebrafish. I think it plays if you haven't seen it. So we're getting this really high spatial resolution and indeed cellular resolution of the activity within the zebrafish. So in some collaborative work with Idia Favabil and Ethan Scott, we went and gave the zebrafish dexmedetomidine, which is basically shutting down the locus coeruleus. So and it effectively works as an anesthetic in extreme dosages. And she has these zebrafish in a particular gel. We're measuring the calcium activity and then apply the dexmedetomidine to turn down this locus coeruleus activity chemically. Here's the behavioral data, so measuring how much the fish is trying to swim. In the control case, you can see it has a fixed mean. And in the case of the dexmedetomidine being applied at the 5-minute mark, we see this reduction in motor response. So we're getting a suppression of motor activity. And then on top of this, the zebrafish is exposed to auditory tones every 30 seconds. So this is the global signal of the fish you're seeing in the 3 conditions. So baseline, before anything's done to the fish, then during the dexmedetomidine, and then finally adiponazole is applied, which actually reverses the effect of the dexmedetomidine. So you can see in the global signal that the stimulus response is pretty much conserved. But if we go through and apply, I essentially did a fitting of the calcium response function for each cell, so each of the 40,000 cells of the zebrafish, of which there are 7, I think, for every event. So a lot of fitting going on. You can go through and figure out how strong was that cell's response to the auditory tone, and at what time did it occur? And that's the crucial component. So taking into account delay, the idea that a cell need not respond exactly when the stimulus happened, it maybe some delay following it. And what you find is that in the baseline and ADDI condition, the ADDI is essentially like wake as well. We find that there are responses occur at later times as opposed to the DEX. So DEX occurs when the fish has had its locus coeruleus effectively shut down or turned down. We find that there are the really stereotype responses. Most of the cells sort of co-activate. So you would see that in something like a general linear model where you don't take into account delay. But in the case of the baseline and ADDI, we do see this increase. It's a significant increase in probability for, in this case, it's the third DT, which is about, I think the DT is about 0.75 seconds. So yeah, pretty significant change. And this is localized to certain areas within the fish's brain. Here's another way of visualizing it. Essentially, most of the cells, this is all the fish superimposed because I didn't pre-register anything. So some of the locations of the cells are different for each fish. Basically, you see in the DEX, everyone responds early to the auditory tone. And in the baseline and ADDI conditions, there's many more cells that respond at a later time. A sort of cartoon of way of visualizing this is what I'm showing here. The idea being that essentially DEX has a much more stereotyped response to the auditory tone, right? And the wake conditions have this higher response variability or dimensionality. And in the language of computer science, you would say that this is lower compressibility. There's more information in the baseline and eddy conditions because there are these extra delays going on here, right? You only need one bit, a 0 or 1, to encode whether or not everyone responded together or did not respond. But in the baseline and eddy conditions, you need much more information to capture this delayed response. So this could be indicative of computation, right? The idea that a particular cell is doing something with the information first before it sends it on, and the other cell will have to wait for that computation to occur in order to integrate that information. So that's pretty cool. But we've so far looked pretty much exclusively at the locus coeruleus diffusivity, but what about other diffusely projecting nuclei? As I mentioned at the start, the thalamus has diffusely projecting nuclei. There's actually a full panoply of cell types. It's really awesome. If you're interested, I would recommend you visit some of Francesco Casca's work. He's got some awesome papers, and as well as a textbook as well, which is where these figures are from. But the long and the short of it is that there is, just like I mentioned before, there are these cells that target particular parts of the cortex, but then there are other cells that sort of project everywhere. And in fact, there are these really weird cells that do a bit of a mixture of both, which is a whole nother kettle of fish I haven't tackled yet. So basically, I mean, in this work, we were kind of really trying to double click on this core matrix dichotomy, this targeted and then this diffuse. In some work we had in Neuroimaging 2020, we took it turns out that these cells have different calcium binding proteins within them, and you can use genetic data sets to infer for each sort of region of the thalamus how much of each cell type it had. And then you could map that thalamic cell expression gradient or map to the cortex using functional connectivity, which is what we've done here. So you see that frontal areas, a little bit of parietal and some cingulate have this high expression of these matrix cells. Well, they're receiving a lot of input from areas of the thalamus that have a lot of these diffuse matrix cells. And then while we were thinking about these two cell types, we came across these awesome thalamic stimulation works by two separate groups. There's about 3 or 4 papers actually now on it, where they put an electrode into the central part of the thalamus in macaque monkeys, while the monkey is rendered unconscious via propofol. I think in both studies they used. So an anesthetic has been applied, the macaque is unconscious, and they electrically stimulate the central thalamus, and the monkey wakes up. In the Michelle Redenborough study, they showed they have 2 ECOG electrodes placed on the cortex, and you find that there's this recovery. Well, first, propofol destroys a peak in coherence. So there's a 10 hertz oscillation that's humming along, this sort of alpha oscillation in the macaque's cortex, and it's coherent when the monkey is awake. So there's a fixed lag between the peaks, or fixed phase relationship between the peaks of that oscillation for those 2 areas. And when you apply the anesthetic, that coherence is lost. And at the same time, when they stimulate the thalamus of the macaque, they see a moderate recovery of that coherence peak. And then in Jordi Tasseri's work, they also recorded fMRI, and they looked at the correlation structure of the monkey before stimulating the thalamus. So this is when the monkey is under anesthetic, and then during the stimulation, you see this really rich integration. Zero lag correlation increases sort of everywhere. All right, so then the question was, can we incorporate this information, all of these moving parts, into a large-scale biophysical model? And does the thalamic heterogeneity provide insight into how the stimulation that they're applying to the thalamus is modulating the consciousness of the macaques? To do this, we needed to build a slightly more complex neural mass model. So the main addition we've added is this non-specific diffusely projecting matrix population. Now I'm not going to run the model on a network defined as a torus. I'm actually going to combine 2 network components. So here I'm going to use distance dependence. So the idea that neurons' connection probability drops off as a function of distance, as well as white matter fiber density So I combined two of them and I ran the optimization there. And essentially, I'm setting up an axis of variation or a core matrix gradient within the model, where some parts of the system are predominantly isolated corticothalamic loops, and in other parts, it's much more of this, I'll send my signals everywhere, based upon the core matrix gradient that I showed here. So on the right. So we can run this model. We can get some simulated data, some correlation structure, which is moderately comparable to what we see empirically. I also have to model propofol in order to recapitulate the macaque results. So I need to propofolize my model. To do that, I do something pretty vanilla, which is just prolong the inhibitory postsynaptic response potentials, which is one of the things that propofol does. And it's pretty straightforward to apply that to model just by changing some timescales and doing some normalization. I'd also like to pause here and go on a very small tangent, which is to say that in the model, I pitted white matter and exponential distance or distance dependence rule against each other agnostically. I just said, you know what, optimize my fit to functional connectivity data. You can play with a parameter that, you know, might favor one or the other. And what I found was predominantly the optimization favored the distance dependent rule. So the white matter is still included here. So these little hot zones are basically saying this is another parameter that I was playing with to do with the decay rate. But essentially, most of the fit, the good fit that you see to functional connectivity data is driven by an exponential distance rule, and then the white matter contributes a much smaller percentage of that, which is nice because we do in fact see distance dependence dominate connectivity in the brain. And this relates to other parts of physics and indeed engineering and applied math, where we have distance dependence, like vibrational modes in a string. Each of the atoms within the string have a spatial rule associated with their connections, right? They connect to neighboring atoms, but not to atoms at the end of the string directly. So that distance dependence is important. And if you take into account, if that distance dependence rule is at play, you can calculate what's called a geometric eigenmode of the system. It turns out to be a great way of capturing large scale dynamics in the system. And in fact, it provides an intuitive explanation about how the activity is organizing. There's an awesome work by James that's come out recently. And then also we had some work in Frontiers, sort of double clicking on it. It's a beautiful analogy. Well, it's not even an analogy. It's a beautiful way of viewing the system, I think, because it has a lot of overlaps with other disciplines. And when I show a music example here, but it's not just an analogy. Like the reason that we use the music is set up the way it is because the physics of a violin string has a nodal structure to it, which are defined as eigenmodes of the string. Those are the notes, so like a C, an E, and a G. And then you combine those notes together to form a chord. And that might be something like what we see in a principal component, where you're combining together multiple notes to produce a principal component. And then in music, you have motifs and you have bars and all that sort of jazz. But it turns out to be a really intuitive way to view what's happening in a piece of music, right? You don't just sit there and think, oh, there was a wiggle there and a wiggle there. You can go through and say there was a C, E, G, it followed this format. And to really hammer that home, here's what happens if you view the dynamics of the violin string from the perspective of an ROI, where you need lots of wiggles and you're trying to relate these wiggles to each other, maybe with correlation. If you view it as a modal amplitude story, something really nice and simple. You just plucked the system, you got energy deposited in these modes, and it decays in time. Anyway, that's my side point over, my tangent over. Back to the whole thalamic stimulation story. So we have this model, we've propofolized it now. It's got space and geometry of the brain baked into it. And we want to explain this empirical result in macaque. So we went through and we stimulated the matrix population for different parts of the brain. So parts that had a lot of this matrix, these matrix cells, and parts that did not, which I'll call high and low matrix. And what we saw was in the wake condition, we have a high alpha coherence, it's 10 hertz peak, and it's destroyed by propofol. When we stimulated high matrix areas, we saw a recovery of that coherence, whereas it was much less so when we stimulated areas that had small amount of matrix. We see this recapitulation of the integrated correlation structure that they observed in macaque. There's a bunch of different ways you can carve this up too, depending on what sort of answers you like to see in your data. But obviously the coherence result I showed, things like temporal complexity of your time series increases. network integration goes up, time scale, synchronization variance. So the idea that your brain's jumping between being really synchronized and then desynchronized. I think it's a super important measure, and for a flexible and adaptive computational system. Because I'm a physicist, I love to play around and prod the bear a little bit. So what I did was, again, stimulate different parts of this model, different parts of the network. And this is just what the evoke response function looks like for a region when I stimulate it. It's a difference. So I run the simulation first with no stimulation, then I run it again with the stimulation. I just subtract the time series because they have the exact same noise sequences. You can do this, and you can show the nice clean evoke response function. You can look at that response as a function of the wake and propofol conditions, and you find that the wake has this traveling wave, which is what you're seeing here as this black line. So it's propagating in the system, and then you get these rebound ripples as it bounces off the thalamus. So it goes down, hits the thalamus, it comes back up, and these are separated by the time scale of the thalamus. So this will have about a 10 hertz periodicity. In the propofol case, it's way more diminished. These have been normalized, so in fact, the amplitude of the propofol is like more than an order of magnitude smaller. You can go through and ping areas of this is just the cortex flattened out now into a flat sheet. You can ping a part of the cortex that has a lot of matrix and a part that has not a lot. So this would be a sensory motor area. This would be like some frontal area. And you can see time to peak is shown here. So this is giving you how the wave is propagating when I've stimulated the system. And yeah, it's spatial. So we're getting just like a ripple on a pond, it propagates outwards. But then we get these little islands of early activation. And that's because the white matter fibers that are there in the model, but only in a small amount proportional to distance rule, provide little wormholes with which the activity can quickly propagate through and then start another ripple at a distant location. Another way to carve out what the model is actually doing is through information and using information theory. So the two key measures we looked at here are active information storage and information transfer. Don't worry too much about this. Essentially, it's just showing all of the nodes and how much relative to the network, how much they're in a storage mode or how much they're in I'm sharing my information with the system mode. And you can see in the wake and the high matrix conditions, the brain is relatively less in a storage mode compared to the propofol and low matrix. So in the propofol and low matrix, you're kind of doing your own thing. You're not really listening to what the network is doing. You're happy just maintaining your local dynamics. And that relationship is reversed for the transfer entropy. So in the propofol and low matrix, so non-effective stimulation, the system is not transferring information around. That's because it's in a storage mode, and conversely, the high matrix and wake conditions are sharing information. And then going all the way back to that energy landscape calculation I showed you in fMRI, we see the exact same relationship. Here I'm showing the propofol condition and relative to the wake. So you can see wake is much flatter, the energy landscape. I've just taken a slice of that landscape I previously showed you. And the propofol is much more likely to maintain rather than sort of reorganize as opposed to the two wake conditions, which is nice because this story is starting to get really consistent now. And then I just want to finish up with this last point, which is we looked at core matrix, these two cell types. We mapped them to the cortex, and we did this by we're taking the gene expression data that we had for the thalamus, mapping it to the cortex using human fMRI data. So as our transformation matrix, we built a model, we stimulated it, we woke the model up. But this was all in humans, for lack of a better term. We've inferred all this information from what we know about the human data sets that we had. But what about non-human primates, macaque? We looked at the consciousness modulation data in macaque. What about other non-human primates? And this is awesome work that came out by Francisco Casca and led by Paula Marias a few days ago, actually, where they were really interested in a similar question about how the central thalamus is modulating consciousness. And lo and behold, they went in and tested whether what types of connections, thalamocortical connections, do they see in the parts of the thalamus that were waking the monkeys up. And lo and behold, it is in fact these diffuse connecting nuclei. And as he says here, they innovate an array of high-level frontal, posterior, parietal, and cingulate cortical areas, which is awesome. And indeed, some populations simultaneously target both the frontal, parietal, and cingulate cortices. So again, this whole story is really starting to click together. And this is nice because we had somewhat indirectly inferred it, and now there's a direct empirical recording of structural connectivity that's making a similar claim. All right, and then on that wonderful and weird note, I'll conclude. So hopefully I've convinced you that viewing diffuse coupling is acting something like a temperature dial in a thermodynamic system and pushing the system towards this critical boundary where it gains all these extra features of, we looked at things like response time scales, heterogeneity, information, susceptibility to stimuli. We've shown that the diffuse coupling can modulate, attractor landscapes. It forms quasi-critical states. I'm not putting all of these things out here to try and say, like, the idea being that there's so many different ways of viewing the system and what it's doing, and if you can frame it in many different ways, and by framing it many different ways, you really start to understand the object rather than just picking a measure. And I showed a measure changed. I want to show how different perspectives, be they from computer science, physics, neurobiology, neuroscience, imaging, they all roads are leading to Rome here and giving us a nice intuitive feel for what these types of connectivity are doing. And with that, I will end with a wonderful thanks to Mac Shine, Brandon, and the rest of the Shine Lab, and then all of our wonderful collaborators who've been awesome helping us shape the story. And hopefully I didn't give you the illusion that this thing is home and hosed. This was really just the first step. We just trying to make a claim about to a first order what these types of connections could buy you, but there's so much more for us to do, and I'm excited to tackle it with my collaborators in the future. Thank you.